Welcome to the molecular basis of inheritance part 2 of the class. Hope your preparation is going well. And this is the time, the last 20 plus days is left, which is very crucial. So make sure you gather all your strength. Yes, you'll be tired, but motivate yourself. Okay, your goal is very important. And my sincere request to all the dropper students, please, okay, give the full length test. If you're not giving full length test, one doesn't know where you're in terms of your preparation. And post the exam, just take 10 or 15 minutes break and do the exam analysis. If you don't do that either, a part of is left over. That means, okay, your exam is not completed unless and until you cover your question paper analysis. So it's very important. And as far as PW Hyderabad is concerned, from 15th of this month, we're going to conduct a full length test every day in the afternoon session. So please do avail this opportunity, and we are making sure to stimulate the environment in your exam centers. So 2 to 5 p.m. and I've asked the exam cell department not to even switch on the air condition during that period of time, so that proper exam center wala feel aa jana hai. Clear? So without any waste of time, let's get into the class. As I told you, this is a very condensed form of the chapter. So next topic is transcription. So as you know, once the replication part is done, we move on to the transcription, where not the entire part of the DNA is transcribed, not the both the strands are not transcribed. In replication, both the strands of the DNA acts as a template. Okay, over here only one strand act as the template. Even in that strand, the entire strand is not copied. It all depends upon which particular gene has to be transcribed. Only that sequence of the DNA will be transcribed. Okay, so let's see the diagram over here, which is our transcriptional unit, okay, in which we can divide this transcription unit into two different parts. One is your structural gene, one is your structural gene, one is our structural gene, the other one is our regulatory sequences. So there are two sequences. One is our regulatory sequences, where our promoter and terminator are present. So if you ask me, so where is promoter and terminator present? As you can see in the diagram over here, promoter is flanking on both the strands, so as the terminator. But where is it present? Is the promoter present at the 3' end, or the promoter present at the 5' end? So to sort out this problem, in NCERT it is categorically said that everything in the transcription unit is considered in perspective or in terms of the coding strand. So based on the coding strand, we say where the promoter is present and terminator is present. So the promoter is present towards the 5' end, and the terminator is present towards the 3' end of the coding strand. And in the NCERT, Baba clearly says that it is not just these two which acts as the regulatory sequences. There are many other regulatory sequences which are present both upstream and the downstream of the coding strand. Makes sense? See, these are not the only sequences which regulate the transcription. Apart from this, there are many more sequences which controls the transcription, which are located both upstream and downstream. Why am I specifying this? Because statement based questions, assertions and reasoning, if you see a statement where they say promoter and terminator are the only sequences which acts as regulatory sequences, then it would be wrong, because in NCERT it is clearly mentioned that there are many other regulatory sequences both upstream and the downstream. Okay, let's dive into the process. Okay, let's dive into the process and see how actually it is run. So as you know, the process of transcription can be divided into three parts, that is initiation, elongation and termination. So initiation. So who participates in the initiation of the transcription? As you know, transcription is carried out by an enzyme called as RNA polymerase. RNA polymerase is the main enzyme which carries out the transcription, but there is a specific factor called as sigma factor of RNA polymerase, sigma factor, which recognizes the promoter. It recognizes the promoter. So imagine this is our, where is it, sigma factor over here, which recognizes the promoter sequences, binds to the promoter, and then calls the remaining subunits of the RNA polymerase onto the promoter site. See what I'm trying to say? It is the sigma factor of the RNA polymerase that comes and binds to the promoter region and then calls the remaining subunits and adds roughly about 9 to 11 nucleotides and leaves the initiation site of the transcription. Clear? So the main function of the sigma subunit is to initiate the process of transcription, and then the remaining enzyme, which we call it as core RNA polymerase, carries out the elongation process. The core RNA polymerase, except the sigma factor, will carry out the elongation process. Coming to termination, here you need to remember 2 points. That is, there are two ways of transcription termination. That is row dependent. This is a row protein. So row protein dependent and row independent. Row independent. That means without the help of a row protein, transcription will be terminated with the help of hairpin structures, which is not there in NCERT, so I'm not going into the details of this. Okay, so what do you want to remember over here? Okay, the coding strand. Okay, in context of the coding strand, we talk about the promoter and the terminator. In transcription, only one strand acts as the template, that is the strand running from 3' to 5' act as a template, and the sigma factor is the one which recognizes the promoter sequences, and thereby calls the remaining subunits of the RNA polymerase. Elongation is mainly carried out by the core RNA polymerase except the sigma factor. The termination is carried out by two means, that is with the help of a protein called as rho protein, that is rho dependent termination, and the other one is your rho independent termination with the help of a hairpin loop like structures. So in case of prokaryotes, there is only one RNA polymerase, simply called as RNA polymerase, which will transcribe all the three kinds of RNA, that is mRNA, rRNA and tRNA, whereas in case of the eukaryotes, there are three kinds of RNA polymerases. There are three kinds of RNA polymerases called as RNA pol 1, RNA pol 2, RNA pol 3, which transcribes the rRNA, mRNA and the tRNA. rRNA, mRNA and tRNA. Apart from these RNAs, they also transcribes the 28S, 5.8S and 18S rRNA. So it's generally speaking rRNA, mRNA and tRNA, where in case of eukaryotes you have these three kinds of RNA polymerases. So they specifically transcribe 28, 5 point and 18S, and mRNA, that is not exactly mRNA, the precursor form of the mRNA called as the hnRNA, heterogeneous nuclear RNA, which post transcriptional changes it becomes mRNA, and pol 3 transcribes our 5S rRNA and the tRNAs and the small nuclear RNAs. So in case of prokaryotes you have only one RNA polymerase, which will transcribe all three RNAs, rRNA, mRNA, tRNA, over by only one. Whereas in eukaryotes, we have three different types of RNA polymerases. So let's see. Let's see the differences between the replication and transcription. And I don't know, I have this strong feeling that this year, okay, you might see questions by comparing transcription and the replication as well. So where does the replication occurs? Rather, when does the replication occurs? As you know, replication occurs during S phase. During S phase. Obviously, I am talking about eukaryotes. And where does transcription occurs? Predominantly at G1 and G2 phases. I'm not denying it doesn't occur in S phase. It does, but it predominantly carries out in the G1 and G2 phases. During replication, both these strands acts as template. Both these strands act as template. Over here, only one strand acts as template, and that too not the entire strand is not copied, depending upon which particular sequence, okay, has to be transcribed, only that part will be transcribed. Okay, and who carries out the replication process? It is our DNA polymerase. DNA polymerase, which is called as DNA dependent DNA polymerase. This process is carried out by RNA polymerase. RNA polymerase, which is called as DNA dependent RNA polymerase. DNA dependent RNA polymerase. DNA dependent RNA polymerase. Next, it add nucleotides only in 5 prime to 3 prime direction. Only in 5 prime to 3 prime direction. So as RNA polymerase, it adds nucleotides only in 5 prime to 3 prime direction. 5 prime to 3 prime direction. Next, which one acts as a substrate? What are the substrates? The substrates are DNTPs. What are they? DATP, deoxy adenosine triphosphate, deoxyguanosine triphosphate, deoxy CTP, TTP, and so on. Whereas in case of the transcription, the substrates are ATP, or rather I will call it as ribonucleotides. Ribonucleotides, which is your ATP, GTP, CTP, and TTP. Then you'll be absolutely wrong, because in transcription we are making RNA, so it should be UTP. Clear, guys? So this is how you need to prepare, okay, for your exam. Now that you know the lines of the NCERT, but now you need to foresee the questions. While you're reading NCERT, a particular topic, what are the questions that you can foresee? If you can foresee the question, if you can think a topic in terms of a question paper setter, then you're all, like really prepared, and you're good to go for your exam. And that's how the attitude of the student has to be, not simply mugging up the lines. If you are seeing a particular paragraph, what sort of questions can be asked? If you can imagine the questions, if you can visualize how questions can be framed from that particular topic, it is done and dusted for you. Next, okay, I would like to bring, share the focus onto the terms called as gene. What is a gene? What is gene? Gene is basically, okay, a functional segment of the DNA, right? But it controls a character. Usually that's how we have learned the definition of gene in the genetics part. But a gene can make a mRNA, which will be later translated into the polypeptide chain, and that polypeptide chain govern a particular trait, or I would rather call it as a character. It's better. But a gene can also make rRNA. A gene can also make tRNA. But these two do not code for any protein. Do not code for any protein. So, if I call the segment of DNA which is coding for mRNA as a gene, and in turn which controls a character, and the segment of DNA which is coding for RNA and tRNA as a gene which doesn't code for any protein, that means somehow I need to introduce a new term to call that gene which can code for mRNA. Clear? Hello? The sequence, imagine this is the sequence of the DNA and this is a sequence which is coding for mRNA and this is a sequence which is coding for tRNA and this for rRNA. If these two are not coding for any protein and this is the only one that is coding for protein, in molecular basis we need a new term to differentiate between the gene which codes for mRNA and the genes which codes for the tRNA and rRNA, and that's how we ended up with a term called as cistron. Cistron is that gene which codes for a particular polypeptide chain. And there are two different types of cistrons, that is your polycistron and monocistrons. Where are these polycistrons seen? These are seen in prokaryotes and these are seen in eukaryotes. So genes are located on the DNA, which is a functional unit of inheritance. DNA sequence coding for tRNA or rRNA, okay, are also defined as a gene. So in order to bring the differentiation between these three, okay, scientists have introduced a new term called as cistron. So cistron is a defined segment. Cistron is a defined segment, defined as a segment of DNA coding for a polypeptide, and there are two types of cistrons, that is polycistron and the monocistrons. Okay, what do you mean by polycistrons? Many genes, okay, that works in a controlled coordinator manner and a common promoter and a terminator is your polycistron. That means many genes work together as a single unit. So the structural gene is monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacterium or prokaryotes. In eukaryotes, the genes are split gene concept. So as you know, in case of eukaryotes, we have the split gene concept. Split gene concept. Okay, what do you mean by that? Okay, what do you mean by that? Imagine this is your DNA, eukaryotic DNA. So ours is divided like this. There will be a coding sequence called exon and intervened by a non-coding sequences called as introns. So this is called as the split gene concept. And this is what Baba says as a reminiscent of antiquity. Reminiscent of antiquity. Not many of you, okay, gone into this line. This line is very much of NCRT. Reminiscent of antiquity. Antiquity matlab Purana. Okay, you know the antique furniture, antique jewelry. That means which is there, for, okay, in the past. So reminiscent matlab yaad dilana. So this presence of this non-coding sequences introns is a reminiscent of the antiquity. So make sure you or get up for such kind of questions as well. Clear? So next is the next topic is the post transcriptional changes. Post transcriptional changes, which is also called as mRNA processing. Why this is required? In case of prokaryotes, imagine this is a cell. There is no nucleus. So if this is the DNA, and this is your mRNA, and even before, so this is your entire length of DNA that is being transcribed. Even before the entire length of DNA is transcribed, only this much of mRNA is made. Yet this portion has to be copied, transcribed, but the mRNA will be soon translated. Why? Because it's happening very much in the cytoplasm, where you can see nucleases. The nucleases might chop the mRNA. So in case of prokaryotes, transcription and translation are coupled. That means there is no gap between transcription and translation. They occur soon after. But in case of eukaryotes, imagine this is a cell and this is our nucleus. And this is our DNA. And this DNA is transcribed into the RNA. Now this RNA is not ready yet. So this is a nascent RNA which is just being made, which we call it as hnRNA. After undergoing changes, it will travel into the cytoplasm onto the ER, where translation or our protein synthesis occurs. So if it has to travel from nucleus onto the ER in the cytoplasm, there has to be a vehicle, isn't it? It can't just abracadabra ante vachedi kada. There has to be a medium, there has to be a source, so it has to swim. So in order to swim, it needs its tail, and the ends have to be protected as well. So for all these reasons, the hnRNA which is just being made undergoes several changes like 5' capping. 5' capping, that means the 5' end will be protected by adding a modified base called as methylguanosine. Just like a hat, ఒక టోపీ లాగా ఇది మన 5' end అనుకుంటే ఇలా యాడ్ అవుతుంది అనమాట. దిస్ ఈస్ అవర్ హెచ్ఎన్ఆర్. ఆ యాడ్ చేయడం వల్ల బరువుకి ఇలా బెండ్ అవుతుంది, సో దట్ ద 5' end is protected because the ends are prone for nucleus digestion. Imagine you are walking Okay, right now this is summer, you're walking barefoot on the road. Obviously what? You'll get burnt. So you need to have a hat or an umbrella, which is our 5' end, and our feet, which is our 3' end. So you need to have a proper footwear. So the ends have to be protected, and the 5' end is protected by adding a modified base called as methyl guanosine. Okay, and the second step is your slicing. Slicing of what? Slicing of unnecessary things. And you should also do the slicing right now. Okay, stay away from your mobile, stay away from gossiping, stay away from those friends, okay, who brings you down in terms of your energy. And there will be a certain friend who calls, or yaar mujhe tension ho raha hai, okay, I'm under stress, I'm under anxiety, and they pass on their stress on to you. You better stay away from those people. Clear? So you need to slice off what? Slice off the introns. So as I told you, in case of eukaryotes, you have a split gene concept, right? So this is exon, this is intron, exon, intron. So you need to slice off the introns. Okay, you need to chop off the introns, and then later the exons will be joined together. Next comes your third one, that is addition of poly-A tail. A matlab, don't think, okay, only adenine. No, actually it is ATP. Roughly about 200 adenylate molecules are added, roughly about 200, which provides energy so that it can swim from the nucleoplasm into the cytoplasm, thereby on to the ER. Am I clear, guys? Okay, so in case of eukaryotes, after transcription, the RNA is not quite ready to undergo translation. So we call that as a hnRNA, heterogeneous nuclear RNA, which undergoes several changes to become mRNA. What are those changes? Addition of a cap. Where? To the 5' end. Through what? The hat is made of what? Methyl guanosine. It's not just a guanosine, it is a methyl guanosine. And chopping off the introns with the help of a lysosome, which is a protein complex. Okay, see here, these are your introns that are lariat, okay, that are removed. Okay, and then the exons are joined together, obviously with the help of ligase, and there you go, your mRNA is now ready. Am I clear, guys? Hello? Are you with me? And some students ask me, so what is the sequence of these 3 steps? Which one happens first? And again here, Baba is Baba. Baba in the paragraph, okay, talks in a different order. Baba in the diagram talks in the different order. So in that case, what do you go with? I will give importance to what is written in the text rather than the diagrams. So I'm sure you don't ask, but I'm telling from my side that there is no order that is given clarity in terms of NCERT. In the text over here, it's in a different order. In the diagram, it is in a different order. Hope you have seen and realized the small change within your NCERT. Okay, let's move on. Next comes your genetic code. Okay, that big bada table. Okay, do you want to learn? Should we learn? Yes. Now that choice is gone, every line, every diagram is important. I'll just give a small shortcut how to remember the table so that you can pick it up as well. Before going into that, it's not just the table, but there are 4 scientists that are associated with the genetic code. Let's see who are they. The first and foremost one is George Gamow. Who is he? George Gamow, who suggested that who suggested that genetic code should be triplet. Genetic code should be triplet. Why? As you know, there are 4 nucleotides or 4 bases, rather I'll call 4 nucleotides. And how many amino acids are there? 20 amino acids. If each one codes for 1 amino acid, okay, if each one codes for 1 amino acid, with these 4 nucleotides, I can code only for 4 amino acids. So how many are left over? I can't code for 16. So genetic code can be anything, but it can't carry 1 nucleotide. So this is ruled out. If genetic code is doublet, okay, this is singlet, not possible. If it is a doublet, that means 2 nucleotides working together. We have 4, okay, in 2 combinations. You can code only for 16 amino acids. Why am I saying this? Question can be also asked, what if genetic code is a doublet? How many amino acids we can code? We can code only for 16 amino acids. See here, sometimes you need to read the lines in between as well, beyond and above. What if genetic code is doublet? If genetic code is doublet, all we can code with the 4 nucleotides is 16 amino acids, and 4 amino acids will be left over. So we can't code for genetic code can't be a doublet. If genetic code is triplet, that means you'll get 64 codons. And how many amino acids we have? Okay, there are only 20 amino acids. Well, more than required, it's fine. And that's why we call genetic code as degenerative or degeneracy. Clear? So the first scientist is your George Gamow, who suggested that genetic code is, or rather should be, triplet. The other scientists are Hargobind Khorana, okay, and Marshall Nirenberg, and Severo Okawo. Okay, so these 3 scientists are very, very important as well. What is the contribution of these 3 scientists in deciphering the genetic code? The term that I am using is deciphering, decoding. So who are the 3 scientists? It is Hargobind Khorana, okay, Marshall Nirenberg, and Severo Okawo. Hargobind Khorana, okay, is the one who has artificially synthesized the nucleotides in terms of the homopolymers and copolymers. Okay, homopolymers and copolymers. What do you mean by that? He has developed a process to synthesize nucleotides like this. Does it make sense to you guys? Hello? This is your homopolymers like this, or even in terms of the copolymers like this. Okay, this can also be a potential question. Next comes our Marshall Nirenberg, who has developed a cell-free system for the protein synthesis. Cell-free system for protein synthesis. And then comes our Severo Okawo, who has identified an enzyme called as polynucleotide phosphorylation, through which, okay, we can make an mRNA sequence. So thanks to Hargobind Khorana, he made what? A process to make the artificially nucleotides in terms of homopolymers and copolymers. And then Severo Okawo, okay, has identified an enzyme called polynucleotide phosphorylase, or simply you can call it a Severo Okawo enzyme. And finally, Marshall Nirenberg, without the help of a cell, okay, he developed a process, how to synthesize proteins. There you go. All you got all the ingredients. So based on these four scientist contribution, we have finally deciphered the genetic code. We have finally deciphered the genetic code. Fusion. So as you know, genetic code is triplet and genetic code is unambiguous and specific. Unambiguous and specific, and there is no ambiguity. No ambiguity. For I'm telling you, for those students who don't know what do you mean by ambiguity, ambiguity matlab confusion. So what is the confusion, sir? Are bhai, wherever AUG is there, wherever UUU is there, it codes only for a particular amino acids called as methionine, and GUG which codes for valine. Whether AUG is in the first position or 100th position or 1000th position, it codes for only one amino acid, that is methionine. And whether it is a heart cell, a kidney cell, or a donkey cell, or a monkey cell, or a plant cell, or a bacterial cell, it codes only for one amino acid, that is methionine. That is what means there is no ambiguity and it is specific. And it is degenerative. All we need is 20 codons to code for 20 amino acid, but we have more than required degeneracy. And genetic code is read in a continuous fashion. There is no punctuation, no comma, no period, no exclamation marks, and it is nearly universal, and that's the reason why genetic engineering is possible, and that's the reason why we're able to synthesize insulin through E. coli. E. coli doesn't need insulin, which is a human protein, but we are making a bloody prokaryotic cell to make a human protein. That is possible only because genetic code is universal. This is a potential question as well. What do you think that made possible that a prokaryotic cell like a E. coli is capable to produce insulin through genetic engineering? Is because genetic code is universal, and which also says in a sense for all the living organisms we might had a long time a common ancestor. Hello, see, and next the important one is out of this 64 we have 61 which acts as the codons, and the rest of the 3 are the stop codons. In this 61 codon there is a particular codon called as AUG which acts it has got dual function. It acts as dual function, that is, it acts as a start codon, okay, and it also adds an amino acid called methionine. What kind of amino acid is methionine? Anybody? What kind of amino acid is methionine? Anybody? What kind of amino acid is methionine? Anybody? Sulfur containing amino acid. What is the other sulfur containing amino acids? Hello? What is the other sulfur containing amino acid? That is your cysteine. So methionine and cysteine are your sulfur containing amino acids. This is your genetic code. One should learn. If you ask me, yes. Now you can't leave anything. If you haven't learned, okay, there is a small trick. First one is you write the empty table and then mark your start codon, and then mark your stop codons, which are 3 of them: UAA, UAG and UGA. These are your stop codons. I'll tell you, this is the only place, or the second one, where I use menomics. Otherwise I don't use menomics at all. Okay, my students in offline classes, they know that I don't use menomics. Okay, very rarely I use. Okay, because why? There are so many YouTube, even students are also by hearting menomics, but there's so many menomics. You need a menomic to remember the bloody menomics. So I don't really suggest menomics for everything, but here it demands for one such menomic. I'll just give one menomic for this row, and for the rest of the 3, based on the amino acids, you can come up with your own menomic. For the first one, as you can see, you have phenylalanine. Even though it is written as P, the phonetic sound is F. And even the single letter abbreviation is F as well. And then comes your leucine, which is L. So these 2 codes for phenylalanine, these 2 codes for these 2 codes for leucine, and these 4 codes for leucine as well. These 3 codes for isoleucine, and these 4 codes for valine, which is V. So the menomic that I'm going to give for this is, it's a little bit funny, but this is how we can remember: FLIV. So how can we remember this? This is how you can remember. First, love is my Valentine. It's a fun way to remember. So if you remember like this, you can develop your own menomic. For example, I can use a different menomic for this. All these 4 codes for 1, all these 4, all these 4, all these 4. What are this? Serine, S, proline, P, threonine, T, alanine, A. So the menomic for this second one is septa. Serine, proline, threonine and alanine. So this is how you can develop your own menomics. The other way of remembering, at least, so I can't remember so many of them, but at least I can remember like 5 to 6 of them. What should we remember for sure? Obviously, one should remember AUG, one should remember the stop codons, and one should remember identify the codons which codes for aromatic amino acids. So if you expect difficult questions from this topic, this is how one can ask. But before, okay, you give the answer, one should know what are your aromatic amino acids. Tyrosine, tryptophan. So if you can remember like this, okay, you can remember few codons, or name the codons, okay, that codes for the amino acids which forms the nucleosomes. Then you need to recollect, which are included in nucleosomes. What are those? Your lysine and arginine, basic amino acids which are over here. Clear, guys? Hello? This is how one can, okay, remember and recollect this particular topic of molecular basis and inheritance. Okay, one should remember, okay, even this part, especially, okay, this 28S rRNA and 23S rRNA. Why am I specifying these two? Why am I specifying these two? You might have studied something called as ribozyme. What is ribozyme? RNA that is acting as an enzyme is called as ribozyme. So this 23S rRNA and 28S rRNA can act as a ribozymes. What does they do? They add peptide bonds between two amino acids during translation. Obviously 23S rRNA is present in prokaryotes, whereas the 28S rRNA is present in eukaryotes. Okay. So this is the summary of your transcription process. Okay, this is what I was looking for. So the entire thing is there. I think I have gone through the entire details of the transcription, but if you want to recollect, okay, you can take a screenshot of this particular one where the entire process, transcription is embedded into one single slide. Next comes our translation. So the first and foremost one, as you know, is the entire process can be divided into 3 steps. Okay, just like replication and transcription. That is initiation, elongation, and termination. But before initiation, there occurs a process called as activation of amino acids. Activation of amino acids, which is carried out by an enzyme called as amino acyl tRNA synthetase. What is it? Amino acyl tRNA synthetase is the enzyme which activates, first one, which activates the amino acids, and the second function that this enzyme carries out is adding the activated amino acid onto the tRNA. Thereby the tRNA becomes charged. Because in this activation of amino acids we use ATP. So when this activated amino acid is added onto the what? tRNA. Where? onto the 3' end of the tRNA. That's when the tRNA becomes charged. Clear? Is the first and the second one clear? So activation of the amino acid by an enzyme called as amino acyl tRNA synthetase, and then addition of this amino acid onto a tRNA, okay, onto a tRNA, where, okay, so imagine this is your, okay, 3' end. Here the activated amino acid will be added. Next is initiation. Attachment of small subunit of ribosome with mRNA. Okay, that is the first one. This is a previous year question, guys. This is a previous year question. In 1 year, he asked during translation, who comes first onto the site of protein synthesis? Smaller subunit, larger subunit, tRNA, amino acids. It is the smaller subunit that comes first, and then comes your mRNA, and then comes your tRNAs, and then comes your larger subunit. So this is the order of participation. Who? Smaller subunit, mRNA, the initiated tRNA, and the incoming tRNA, and then comes your larger subunit. Okay, so attachment of small subunit of ribosome with mRNA, charged tRNA specific for initiation codon reaches the P site. The first site is called as P site, and the second site is called as the A site. Okay, and the ribosome moves, and the P site becomes the E site, and the A site becomes the new P site, and an empty one becomes the A site. Am I clear? So I would like to ask a question over here. Imagine this is your mRNA. This is your ribosome. Okay. Now tell me, I'll ask a question. What happens? What happens actually during proteasing? Will the mRNA come through the ribosome, or the ribosome glides along the mRNA, or on the mRNA? Give me your answer. Give me your answer. Hello? Will the mRNA pulls through the ribosome, or the ribosome glides along? Obviously, the ribosome moves along one codon at a time. So it occupies 2 codons, and then it moves 1 codon at a time. So it is ribosome that moves along. And what do you have over here? This is a 2022 question. UTRS. UTRS. What are these? Untranslated regions. That means this part of the mRNA is not translated, but they increases the efficiency of the translation. What are these called as? Untranslated regions. Untranslated regions. Agar if these are not being translated, ఒకవేళ ఇవి కనుక ట్రాన్స్లేషన్ లో పార్టిసిపేట్ చేయకపోతే, why are these over here? They increase what? They increase the efficiency of translation. So imagine this is our codon AUG and UUU. So a cognate tRNA, I'm using a term called cognate. It is there in NCERT. Baba says, okay, amino acid will not go and attach to any random tRNA. Again, assertion reasoning, statement based question. An activated amino acid can be added on to any tRNA. True or false? Absolutely false. A amino acid can be added only onto a cognate tRNA. Cognate matlab what? Corresponding or respective. What do you mean by that? What do you mean by that? AUG is codon. Okay, which will add methionine. But to which tRNA? But to which tRNA? A tRNA in which its anticodon should be UAC. For such tRNA, I will add methionine to the 3' end. That's what this statement means. Am I clear? Hello? Am I clear? Let's move on. Okay, lac opron. Lac opron. You need to remember the entire diagram, and many times they ask this sequence. Okay, lac means lac opron. That is lac I, lac Z, lac Y. Okay, and lac A. Z, Y, A. This is I. I doesn't stands for the inducer. The I stands over here for the inhibitor. Okay. Do remember this I is not inducer. This I is for the inhibitor or your repressor or your repressor. Remember, I is not for inducer. Z codes for an enzyme called galactosidase, which convert lactose, okay, into its galactose and glucose. Okay, and Y gene codes for an enzyme called permease, which permits the entry of the sugar into the cell. Like A gene codes for an enzyme trans-acetylase. Okay, trans-acetylase metabolizes toxic thiogalactosides, which are also allowed to enter into the cell by permease. So this particular function is not there in the textbook, but I'm giving it away anyways. So what does it means is, okay, this particular trans-acetylase, okay, it metabolizes certain toxic compounds that enters along with the lactose. Not required. Okay, but here you need to remember, okay, what do you want to remember over here? Okay, this particular lac opron is an example of both positive regulation and negative regulation as well. Okay, in the last line of this lac opron, Baba talks about particular this one. And who acts as an inducer? Who acts as an inducer? Hello? Who acts as an inducer? Okay, in the textbook, it is clearly mentioned it is the lactose or the allolactose acts as a inducer. Okay, it goes and binds to the repressor and brings a conformational change in the repressor, so that repressor can't bind to the operator region. Thereby RNA polymerase can move forward and transcribes a common mRNA for these 3 genes. Okay, and this is a perfect example for operon. What do you mean by operon? Cluster of genes that works in a controlled, coordinated manner is called as operon. What is operon? Cluster of genes that works in a controlled, coordinated manner is called as operon. Okay, and the concept of operon is given by Jacob and Monod. Jacob and Monod. Okay, so these are the few things that you need to remember from the what? Search of genetic material. There is one question I want to ask you. Okay, virulence. What do you mean by virulence? Don't think just virulence is there, it is virus. No. What do you mean by virulence? Okay, that is the disease causing capacity. So, which strain has got, okay, virulence capacity? The S strain. But where does the virulence lies? This can also be a question. The virulence lies in the polysaccharide code. S matlab smooth na. That smoothness comes because of a polysaccharide. This bloody polysaccharide is the one which is causing the pneumonia disease, which is absent in case of the R strain. Hello? Clear? So who is the scientist? The scientist is Frederick Griffith. The year is 1928. 1928. So what is the conclusion? Some transforming principle transformed the heat killed a strain into the R strain, which made the R strain into the virulent strain. Which made the R strain into the virulent strain. Who? R has become S. Okay. This, okay, obviously I talked about in the previous class. That is who proposed the semi-conservative model in an indirect manner. It is our Watson and Crick. But who proved it experimentally? It is Matthew Meselson and Franklin Stahl. Okay, Matthew Meselson and Franklin Stahl in the year 1958. Remember, they haven't used radioisotopes. They have used a heavy and light isotopes of nitrogen. They have used the heavy and light, okay, isotopes of nitrogen. And remember, this 20 minutes is for the entire generation, okay, to see one generation. Okay, if this cell becoming 2, this is generation 20 minutes, but out of which 18 minutes is for replication. Is for replication, guys. Pay attention for all the small details. But Taylor and his colleagues in the same year also proved that not just in E. coli, Baba, not just in E. coli, that DNA replicates in a semi-conservative manner, even in plants. For example, Vishia faber, okay, here they have used radioactive thymidine. Pay attention, pay attention. Okay, but here they have used the what? The heavy and light isotopes of nitrogen. Clear, guys? Okay, and obviously you need to remember the scientists who have, okay, unequivocally proved that, okay, DNA is the genetic material. Who is that? Alfred Hershey and Martha Chase, who has proved that DNA, okay, is the genetic material. Have been asked 3 to 4 times, okay, before. Okay, so thereby I'm going to, okay, call it a day. Okay, very, very important that you go through line by line of the molecular basis thoroughly. I think we covered most of the important points. See, this is how questions can be asked. Okay, if you prepare in terms of the questions, okay, you will not forget for a very, very long time. So please, okay, pay attention while you are preparing 4C questions. Don't simply mug it up. Okay, thank you for watching, and I'll come back with a new video.